DNA assembly is the backbone of synthetic biology, with it being the way we bring our designs to life, but assembly can be difficult. There are many different methods available, each with its own conditions under which it can be used. Our tool aims to remove the decision of which assembly method to use. All the biologist has to do is specify how they want their design to be built, and the tool generates the appropriate protocols. Let's take a look. First, we have the syntax. The user is able to specify an ordered join with an asterisk, which means that the two parts must be assembled in that order. Alternatively, the user can specify an unordered join with a plus sign, in which both parts must be present in the assembly, but their order is not important. Also, the user can place parentheses over parts of their sentence to create a subunit that would be assembled into an intermediate part before dealing with operations outside of the parentheses. Here we have the code open in a Jupyter notebook. Before the user can input their sentence, the cells have to be run to set up the various functions of the tool. This includes the parser, which takes the user's expression and creates a set of expressions and sub-expressions without parentheses. Expressions in parentheses are replaced with placeholder values that are resolved during the next step, which is graph construction. A graph that represents all possible assembly steps is created recursively by resolving placeholder variables based on their join types until the original parts are reached. Now, an expression can be inputted. Let's do open parenthesis a plus b close parenthesis times open parenthesis c plus d close parenthesis. Before generating the protocols, let's analyze what the sentence means in the context of our tool's grammar. We have two sets of parentheses, each containing an unordered join of two parts. The parts in the left set of parentheses could be assembled as AB or BA. Either way, let's give it the placeholder E. Similarly, C plus D could be CD or DC, and we'll give it the placeholder F. Since the parentheses are joined by an ordered join, the subparts must be assembled in the order EF, though the sequence of parts in those placeholders can vary. If we submit this sentence to the tool, we see a series of protocols provided as output. For this example, we use a Gibson assembly protocol. This particular expression generates a series of three protocols, one for joining A and B, one for joining C and D, and one to join the two subparts. Each individual protocol is created using a template of the selected assembly method. The two parts being combined are inserted into the template where appropriate. From our example, we see here parts A and B inserted into the Gibson isothermal assembly template. This is the complete graph constructed based on the example sentence. The protocols are generated by starting at a leaf node and creating a protocol joining the node's two parent nodes. The graph is traversed backwards in this fashion, with each node that is passed getting a protocol that creates that node, until all of the original part nodes are reached. In addition to the set of protocols, the tool also generates a dot file that, with the help of a tool called GraphViz, can be made into a physical graph like this one. The main challenge we faced with this project was with graph construction. Creating graphs that allowed more than two parts to be joined at once turned out to be more complex than expected, and so the tool had to be simplified such that only pairwise assembly protocols are produced. This means that the tool does not take full advantage of assembly methods that allow multiple parts to be joined in one step. We were also unable to implement certain functions into the tool, such as plasmid specification and part protocol compatibility, in which the tool analyzes the parts and determines which protocols are best suited for those parts. Nevertheless, we believe that our tool serves as proof that our concept works, and that a fully implemented tool would be useful to anyone looking to carry out DNA assembly. Thank you.